but you should be safe. Guys, in this mode of transmission, just look at the Indian scenario. In India, percentage of total transmission, 88.9% is sexual. See, 88% is sexual. In India, it's mainly because of sex, vaginal sex, heterosexual. Then is parent to child. Sex and parent to child are the most in India mode of transmission. Unknown surprisingly is higher in India. So remember, in India, the sexual intercourse and parent to child is more, okay? Then moving on in pathogenesis, next let us look at receptor attachment. This is not going to be easy. You have to pay attention here to receptor attachment, okay? In pathogenesis, so much is there, okay? Look at this, receptor attachment, then replication of the virus, immunopathogenesis, kinetics of immune response, and opportunistic infections. We'll cover everything, okay? So it is going to be a long journey. Pay attention here. Now where we are in receptor attachment, okay? Okay, I hope you're awake. So shall we start off with receptor attachment? So you should know the receptors, okay? See, basically the main receptor is the CD4 receptor on T helper cells, right? Helper T cells, there will be CD4, right? So that is going to be the main receptor. So here what you're going to focus on, main receptor is CD4. It is present on many types of cells, like it can be present on helper T cells, monocytes, macrophages, Langerhans cells, astrocytes, keratinocytes, glial cells, and even in dendritic cells, okay, which are in the skin. So it can be present everywhere. So that is the main receptor. Then there is a co-receptor on T lymphocytes. You should have one more receptor here, CXCR4, okay, CXCR4 on T lymphocytes. Wait, we'll draw that also. Bring this diagram here, which is the other receptor, secondary, that is going to be CXCR4, that is on T helper cell. That is going to be our secondary receptor. So remember, CD4 is the primary, that's the main receptor. Then you have CXCR4 is the secondary co-receptor on T lymphocytes. And CCR5 is present on macrophage cells of macrophage lineage, okay? So this much you understood, right? These are the receptors. Then you have something on the dendritic cells. Dendritic cells, they have lectin receptors, but these receptors, no, they won't help the virus to enter the cell. It will just bind to this receptor, and on the dendritic cells, these HIV will attach. But this will help in transport of these virus to lymphoid organs. Did you understand? What exactly are we trying to say in the dendritic cells? The HIV virus bind to these dendritic cells. They don't invade these cells. The dendritic cells help this virus to reach lymphoid organs. That's all. So actually the second co-receptor is there, right? CXCR4 and CCR5. These help the entry of the virus into host cell. So far we saw the receptor binding. This is going to bind, right? Bind to T helper cells, binding over, entry over. Now we will continue with the pathogenesis, okay? In pathogenesis, where we are? Receptor attachment, easy, over. Now we are going to replication. Now the virus has entered, now it wants to replicate. Do we have a slide for replication? Yes, replication. Now replication, what and all headings are there here? Let us see. Fusion. So fusion means the attachment of the receptor and co-receptor to GP120. Fusion of HIV to host cell takes place mediated by fusion protein GP41. So here still they are talking about binding only. Binding of receptors, right? CD4 and co-receptor GP120 of the virus. And now GP41 helps in what? Fusion. GP41 helps in fusion. That's why it's called fusion protein. So you remember GP41. GP41 was actually the transmembrane pedicle. Remember this diagram? In this we have already seen. GP41 is transmembrane pedicle. It's called as fusion protein. It will help in the fusion of all these things. Okay. What and all? The receptors, the co-receptor and receptor, co-receptor and GP120. All these things will bind. Fuse. Now next step here, penetration and uncoating. So basically the virus now after fusion, the HIV nucleocapsid enters the host cell. What is entering? The nucleocapsid is entering the host cell, which followed by uncoating and releases 2 copies of the single-stranded RNA and the viral enzymes. So now what will happen? Nucleocapsid enters, uncoating happens and it releases the 2 copies of RNA and the enzymes. Pretty clear, right? Nobody is confused, right? At this stage, everybody is fine. So where did the virus enter? It entered the cell. Right? It entered the cell. Now the virus is inside, okay? Next step is what? Reverse transcriptase. Now reverse transcription happens. Now reverse transcription is the next step. Now what will the virus do? It has entered. Now it has to convert this ssRNA to ssDNA. So single-stranded RNA becomes single-stranded DNA. Okay. Then, actually this DNA is actually having a DNA RNA hybrid. The RNA gets degraded. Now ssDNA replicates to form dsDNA. Now it becomes double-stranded DNA. So single-stranded RNA became single-stranded DNA. Then further it is becoming double-stranded DNA. Now this double-stranded DNA, what it will do? As you already know, this DNA has to go and join with our DNA, right? But before that itself, some amount of transcription happens and some viral proteins are formed. Then comes the next step. Here, the pre-integration complex. Okay. They're calling it as the pre-integration complex. Basically, it has to go and integrate into our DNA, correct? So this double-stranded DNA which is there has to go and integrate with our DNA. So before that, one pre-integration complex it has formed. The nucleoprotein complex formed, okay, there's a nucleoprotein complex, that is a pre-integration complex which is formed. It consists of what and all? It will have this double-stranded DNA, some gag matrix protein, okay, some accessory VPR protein. All this you studied, remember, in non-structural protein VPR. All this. And viral integrase. Without integrase, how it will integrate? No. So it needs this integrase enzyme. Okay. So this is called as the pre-integration complex. Now it enters the nucleus. Now it is entering nucleus, guys. Now it is entering host nucleus. Okay. Now after it enters host nucleus, the viral double-stranded DNA gets integrated into the host cell chromosome by what? By the viral integrase. Integrates with our host, our chromosome, right? So what is integrating? dsDNA, double-stranded DNA. With help of what? With help of viral integrase, integrase enzyme, correct? Integrase, okay, enzyme. So this is called as provirus. What is provirus? The integrated virus is called provirus. Provirus. What is provirus, guys? Provirus is this double-stranded DNA integrates into the host cell chromosome. The integrated virus is called provirus. Hold on. So we have tried to explain here. This is the helper T cell. This is the nucleus. Inside the nucleus chromosome, this virus double-stranded DNA went and got integrated. This is the provirus now. What is this now? Provirus. Very good. Then there is a latency. That is the last step in this replication we want to explain, latency. Latency is in this integrated state itself, the HIV establishes a latent infection, okay, for a variable period. So many years also it can just be like this only. Okay, so that's all we want to say now in this replication. So what and all we saw in replication? We saw fusion, it will fuse, penetrate and uncoat. Reverse transcription happens, pre-integration complex, enter the nucleus, form the provirus, and then latency. So now that replication is over, the next step is immunopathogenesis. In pathogenesis, you have to study about immunopathogenesis. So shall we go to immunopathogenesis? I think it is too much for now, right? So immunopathogenesis, you have to learn about the acute HIV disease or the acute retroviral syndrome, asymptomatic stage, persistent generalized lymphadenopathy, systemic HIV infection, AIDS. Finally only it goes to AIDS, okay? AIDS is not initial. HIV, then in progression it leads to AIDS. After that we will look at the kinetics of immune response in pathogenesis itself. Then we will see opportunistic infections, lab diagnosis we have to see, lab diagnosis in newborn, clinical diagnosis, HIV, the treatment, all the treatment we have to look at, how to monitor the progression we have to look at, post-exposure prophylaxis like prevention for infection if there is a needle stick injury, right, needle stick or stick. So needle stick injury only it is called. Then vaccination strategy is not important for exam actually. But so far whatever we have seen, World AIDS Day is December 1st guys. So remember at the end of the year only it is. So let us continue next video with pathogenesis of HIV.